Good morning, everyone. My name is Ankita, and in this project, I am interested to express ISG20 gene into mammalian cell via expression vector to determine its effect against antibiotic marker. So, ISG20 is 20 kilodalton protein in the human, which is encoded by ISG20 gene and act as an innate anti-hepatitis B virus effector that effect selectively degrades hepatitis B virus and blocks the replication of infectious hepatitis B virus particle. It is also involved in the number of regulatory functions such as control of cell proliferation, differentiation, and regulation of immune system. So, as we can see here, it is a mammalian expression and a dual tagging PCMB6 entry vector that serves as an entry vector to transform an open reading frame into destination vector using precision shuttle system, having a kanamycin as a selected drug and neomycin as a selected marker. So, moving to the next slide. Here, it is a vector map showing the position of forward and reverse primers and multiple cloning sites. Open reading frame in this vector will be expressed in mammalian cell as a take protein, C-terminal, MYC and DDK tags. I have selected VP1.5 as a forward primer and XL39 as a reverse primer respectively. All of the plasmids in the precision shuttle system allows high-level target gene expression in mammalian cell or via in vitro translation in cell-free system. The plasmid contains the promoter and enhancer of human cytomegalovirus, that is CMV, immediate early gene to drive mammalian gene expression, and the T7 promoter for in vitro transcription or translation process. An optimal causa sequences is included in the plasmid to enhance the mammalian cell expression with attached MYC and DTK tags. So, moving to the next slide. Now, we will discuss the PCR strategy and outline of cloning steps. The open reading frame of the clone must be PCR amplified, as we have selected in order to attach the cloning site to 5' end and 3' end of the sequence. After performing the PCR, confirm the size of the amplification product by agarose gel electrophoresis and purify any remainder of the reaction. Now, we will elute the DNA from the purification column and set a digestion reaction followed by its purification process. Now, we create stable line expression clone and we would perform transfection and later by selecting passage for transfected cells into neomycin growth medium. We will discuss all this in detail in the next slide. So, let's discuss about the transfection and further procedure in detail. Now, we will deliberately introduce our clone vector into mammalian cell, leading to the enhance its gene expression and production of recombinant protein in this mammalian cell. After transfection, do not change the medium until the cells are ready to be passaged, and now passage the transfected cell into vessel containing the growth medium having the neomycin, and its concentration should be determined by the kill curve. After 1 or 2 weeks, if the large number of cells get killed by neomycin, that help us to validate the presence of the correct expression vector. So, in this slide, as we can see here, some validation techniques for the expression vector. I have used CMV promoter and V2F as a forward primer and XL39R as a reverse primer for sequencing. Here we can see the expression of C-terminus fusion tags, which are MYC and DDK tags, can be validated by the Western blot analysis, as shown in these two figures. And we can also validate it by using the verifying sequence we have got here with the sequence information which is available online as a chromatogram graph. Thank you.